Okay. Yeah, I'm not scared. I just think we could do more. Let's just talk about some other key terms here. This could be fun, I think. Kind of like we did before. To me, if I was to focus on vocab at any point in time, I would say it's really through chapter 16 and 17 where the vocab got or became like maybe disjunct. And so I just want to go through some key terms that are in those two chapters. So I'm just going to start. Translation, there's transcription, there's replication, we talked about transformation, there's transduction, let's see, what else is there? There's reverse transcription, let's see, there's methylation. We also talked about acetylation. And while we're at it, we had also mentioned conjugation. So a lot of Asian words. In fact, they're all Asian words. A-T-I-O-N, awkwardly enough. Cool? Okay. So let's look at translation real quick. That's the event at which you make a protein, right? Make protein by combining amino acid. And of course, this is at a ribosome. Transcription is the event in which you are reading DNA to make mRNA. You might modify it, you might not, depending if you're a prokaryote or eukaryote. Replication is an event in which you make new DNA. Right? It represents the S phase. The major enzyme of transcription is the RNA polymerase. The major enzyme of DNA replication is the DNA polymerase. And to me, that's one part that I want to make sure everyone's got disjunct in their head. Transformation, the uptake of DNA from the environment. This is something we see bacteria do all the time. What was the name of the scientist that first saw this? Very good. Griffith. Transduction. Viral DNA into bacteria. We see this with bacteriophages pretty often. Reverse transcription. Literally, it's a reverse transcription seen with retroviruses. Retroviruses take mRNA and create DNA out of it. This is the process that we would see in a retrovirus, like for instance HIV. HIV's mechanism, utilizing the enzyme reverse transcriptase. Methylation, basically to silence the DNA, to shut it down. Acetylation, that's to turn on the DNA. Basically, silencing the DNA would make it unreadable, which means no protein synthesis, and this would actually make the DNA readable, so you could do protein synthesis. And conjugation is when bacteria share genetic information. Bacteria will share plasmids. It's also known as bacterial sex. A mechanism of sharing information between each other. Well, let's continue on. There's a lot of topics that you might have questions on. I promise you there's about 2 hours worth of video that's going up to YouTube already, and this is going to make close to about a third hour. And then there's no telling what we'll do tomorrow. So you have like all these resources to assist you in prepping for this exam. They're really, there's no excuse. Let's talk about doing test crosses. How about the test cross? Let's remind ourselves why we would do this, why this is the case, why this is something we would even discuss. Remember, test cross would help you determine. So it helps determine the genotype of a creature, you could say. And the reason that we would need to do this, the reason we would need to determine the genotype, is because usually homozygous dominant and heterozygotes are having the same phenotype. Again, what this statement is kind of saying essentially is that if you are big T, little t, well, we really can't tell much difference between that and big T, big T. Agreed? They both would appear tall to us. So here's what we do in the test cross. We always mate the dominant phenotype, which of course is going to be one of those guys, with mate the dominant phenotype with the recessive individual. And to me, you would see two outcomes depending on what the original parent was. If you had a parent that was big T, mating them with the recessive is going to create progeny, is going to create babies that are all showing that dominant phenotype. Or if your dominant phenotype was actually heterozygous, then the test cross would reveal that half of the offspring, half of the progeny, would have the dominant phenotype, the other half would have the recessive. Cool. Okay. Right? So this can kind of reveal what you have if you have the dominant phenotype. Hopefully we're 100% on that. I'll move on and let's talk about the hierarchy of organization. Of course, this would be biological organization. Now remember, and this was a question I know you had on your review, it starts out with the atom. And of course, atoms build what? Okay, molecules. And what do molecules make? Okay, I'll just finish it. Organelles. Organelles make cells. Cells make tissues. And tissues make what? Go Mustangs. Organs. I almost wrote Go Mustangs instead of organs. My bad. Organs make what? Systems. What do the systems combine to form? Very good. Organism. What does an organism form? Good. Population. I like the diversity in responses. What does a population form? Okay. Community. Good. Let's keep this going. What's a community form? Okay. Ecosystem. And what's an ecosystem form? Biosphere. Or planet Earth, as we call it. Okay. I wanted to snake it around this way to conserve paper. Very good. We good with that? So from the very small, obviously the atomic part is very small, and the biosphere is of course very large. Duh. An atom is small, planet Earth is huge. In a previous video, in an earlier video, I had talked about the process of non-disjunction. A non-disjunction is an event in which you can end up with an individual having a unique set of chromosomes. So this is actually what can cause monosomy or trisomy, depending on the specific point in time that non-disjunction occurs and how it works out. Of course, it's usually going to be a very, very negative thing. Certainly a negative event. And can someone name me a disease that I could study with non-disjunction? Probably the most common one would be Down syndrome, which is also known as trisomy 21. Now there's a variety of other trisomies, but this is probably the most common, and so therefore probably the most discussed, trisomy 21. There's also monosomy X, and Klinefelters as a result as well. Very good. A lot of sex-linked or sex chromosomes non-disjunction events, but those probably right through there are certainly the most commonly discussed 3 in an AP Bio class or in an intro college class. There's one just about everywhere. It's just that a lot of times, like if it's around certain important ones, like trisomy, something important, chromosome, usually the fetus doesn't develop into birth stages. But yeah, I think like, there's trisomy 8 and they're born, but they're born with like major issues and complications that prevents their life from being any longer than a couple of like years at best. This is kind of thrown out there because this is an individual that could live what I call a normal lifespan. Obviously it's not a normal life, but a normal lifespan for sure, and make it, if you will. So non-disjunction. Let's see. Let's talk about feedback inhibition and metabolic pathways, because they're a topic for sure that's on the exam. So metabolic pathways. Let's take compound A and react it to form compound B, which could react to form compound C. And I could go on indefinitely. I'll go to D and then maybe to E. And then of course each of these is requiring their own separate what? Enzyme. So like let's call this maybe E1, and then there would be E2. Bless you, by the way. There'd be E3, then maybe E4. Good. Cool. And I think what we could see here is that there's going to be different enzymes that are catalyzing different reactions, correct? Now there's two different things that could occur here, if you think about it. We could have a system, we could have a situation in which we speed up the process, and then we could have one in which we slow down the process. So to me, we could have maybe like an activator or an inhibitor of this reaction, correct? And that's going to result in two different events. That's going to result in a type of feedback called positive feedback. And then of course there's the direct opposite, which is going to be the negative feedback mechanism. So again, in positive feedback, what we do is we start up the system even stronger. So this is going to result in a larger production, if you will, of E, which of course is your final result. And in negative feedback, this is going to result in decreased production of E. Yeah, so it works in such a way. And usually the way negative feedback works, because to me that's the one that you would want to spend the most time on. Usually what would be the ability or the cause of this to occur is the simple fact that compound E turns off one of the enzymes, probably one of the first enzymes. So it's likely that the negative feedback mechanism is substance E turning off enzyme 1. Cool. Yeah. Could be enzyme 2, but that would be, to me, unlikely.